Welcome to Random Review. Thank you for joining us. I'm Connie Georgiou. Deborah Goldenberg and I coordinate Random Review for Friends of the Library, which sponsors these events. Deborah, will you join me on screen and greet our audience?
Hi, everybody. Enjoy the review.
And our library host today is Bonnie Brzozowski. She'll be fielding your questions at the end of the program. Bonnie, would you like to turn on your camera and say hello?
Hi, everybody. Welcome.
A few operational details. If you're having any problems with GoToWebinar, please use the question or chat box on your control panel and we'll try to help you. We're recording this presentation, so if your issues can't be resolved, be assured that a link to the recording will be sent in your follow-up letter. Also, there'll be a link to a more polished version on the Random Review page of the library website soon. On the Random Review webpage, you'll find a link to a list of library books related to today's topic, created by our volunteer, Gail Gerdeman. And on the same page, you can sign up to receive monthly Random Review email announcements. We encourage you to submit questions for our speaker at any time during the presentation by typing them into the question or chat box, and she'll answer as many as we have time for at the end of the program. Great news! Friends of the Library's Big Book Sale is returning in just a few weeks. February 25th, 26th, and 27th. We'll be at the fairgrounds as usual, but in the Solar Building this time. Details of the event are available on the Friends of the Library Facebook page. We're still recruiting volunteers to work at the sale. If you'd like to lend a hand, please let us know at the Friends of the Library's volunteer email address, which will be posted in the chat box during the review and will also be included in your follow-up letter to this event. I'd like to thank our community sponsors of the Random Review program, Grassroots Books and Music, Northwest Graphic Imaging, and the Corvallis GT. A big thank you to the Best Library and especially librarians Bonnie Brzozowski and Mike Hansen, who provide indispensable support in the presentation of our virtual programs. Next month's review will be on March 9th, when Marisa Chappell will discuss the book, The Second Founding, how the Civil War and Reconstruction Remade the Constitution by Pulitzer Prize winning historian Eric Foner. And now I'd like to invite Dan Arp, OSU Distinguished Professor Emeritus and former Dean of both the OSU College of Agricultural Sciences and the Honors College, and currently Executive Director of the Agricultural Research Foundation, to introduce today's speaker. Dan?
Thank you, Connie, and thank you for the opportunity to be a part of this random review. It's certainly my pleasure to introduce our reviewer today, Barb Taylor. Barb came to Oregon State University in 1991 and worked her way through the academic ranks as a professor in the Department of Integrative Biology. I've known Barb her entire time at Oregon State University and consider myself fortunate to be able to count her as one of my friends. In fact, Barb and I currently teach a University Honors College course together on tribalism in America. I think Barb is uniquely qualified to review this book for us for a number of reasons. And first of all, her scientific discipline involves studying the behavior of fruit flies, and she used pretty much every genetic technique available at the time to understand how these fruit flies were behaving. I believe that if Barb was currently running a research lab and not in her current professor emeritus status, that she would almost certainly be using the CRISPR technique that she's going to tell us about today in the book that she's reviewing. Second reason I think she's very qualified to do this is, of course, that she's a woman in science, and at roughly the same time period that the star of the book had worked through her scientific career. So I think Barb is in a good position to understand some of the challenges that the star of the book was dealing with during her time. And third, Barb has thought deeply about ethics and morality in science, how we use science, how we conduct it, and so on. In fact, she taught a graduate course on ethics and responsible conduct in research. And those of you who've read the book know that you get into some interesting moral and ethical issues in thinking about how to use this new technology, CRISPR-Cas9. So for all those reasons, I think Barb is uniquely qualified to review this book for us. And without further ado, let's invite Barb to the floor, or perhaps I should say to the screen, to review the book for us. Barb?
All right, Dan, thank you very much for that kind introduction, and Connie for the invitation to present this book. And so I'm gonna, I really appreciate the opportunity, and I hope you will all will enjoy the review. I'm gonna share my screen now, and so you can, and we'll get started. Here we go. So let me see if I can get rid of my view. All right. So one small problem, I am seeing too much of myself here. All right. All right. Okay. So here's, so now you can see, this is the book we're going to review today. It's Codebreaker by Walter Isaacson. And he's really a fascinating character in his own right. He's professor of history at Tulane University. He's been the chair of CNN and an editor at Time magazine. And his goal here is to show an up close and personal view of how science and discoveries happen through the lens of a particular scientist, largely, Jennifer Doudna. And spoiler alert, she's going to be a co-recipient for the 2020 Nobel Prize in Chemistry. And she's going to get this prize along with another woman, Emmanuelle Charpentier, because she's going to have figured out a structure and function relationship for a new tool, CRISPR-Cas9, for gene editing. And at the end, we'll get to how this might impact the future of the human race. And he's broken this book up into 9 segments with lots of little chapters where he does focused presentations and discussions about particular individuals and their contributions to various scientific discoveries and the science itself. So it's lots of small chapters, and it turns out to have quite a large cast of characters. So we'll get to some of those characters, but not all of them. So I'm going to get started. So we're going to start with Jennifer Doudna. She was born in 1964, and she's going to grow up in Hawaii because her father decided after a career as a writer for the State Department to go back to school, get a degree and become a professor of literature, American literature. And the job he got was at the University of Hawaii. And so she got to grow up in this beautiful environment, this beautiful natural environment, a place she loved to be. And in that place, she was exposed to lots of really interesting plants and animals, and she had walks and discovery with her father. And this is a picture of a mimulus. It's a plant that's sensitive to touch, and it's endemic to Hawaii, this particular version. And she found a lot of relaxation in the natural world. She didn't feel quite so comfortable in her community or in her school as she was growing up. And her father really felt his daughter had a lot of potential, and so he pushed his daughter to think broadly and to explore the world. And one of the ways that he did that was to give her books to read. In the 6th grade, he puts this book on her bed. It's The Double Helix. Many of you probably have heard of this or know about this book. It's James Watson telling of the Nobel Prize winning work that he and Francis Crick did for discovering the double helix, the structure of DNA. And in this book, of course, she learns about Watson and Crick from Watson's viewpoint, of course. And he has a particular viewpoint. He is always quite opinionated. And she also learns about a woman scientist, Rosalind Franklin. And Rosalind Franklin is someone that that Watson writes about with respect for her skill at producing X-ray crystallographic images of DNA. They're the images that are going to really help them cement their understanding of the structure of the DNA molecule. But he's somewhat dismissive and condescending about her analytical skills and her appearance and her unwillingness to be more female and feminine in her appearance. And I was really struck by what Jennifer writes about having read this book as a child. She says, "What mainly struck me was that a woman could be a great scientist. The book made me realize you could also hunt for the reasons why nature worked the way it did." So even though at the time she read it, she might have been somewhat aware of how Watson saw Rosalind. And he called her by a nickname she didn't like, Rosie. But it was something that allowed Jennifer Doudna to see that she might have a future as a scientist. And I think that's a really important thing to think about in how we view works that we think is some way might have a different impact on others. And so this is going to be part of her motivation for continuing. She finishes high school in Hawaii. The high school is not very academically challenging or demanding. And her school advisor doesn't recommend going to college because he doesn't think girls need to go to college. But her father pushes her to go to college, and she applies to and is accepted to Pomona College in California. There she's going to get a degree in chemistry, and she's going to work with the only female mentor that she had, which was Sharon Pansenko. And in that lab, she is working with bacteria that have these interesting properties of when they're starved, they form these little collectives, sort of slug-like. And Jennifer has her first real sort of scientific success in a research lab, which is to be able to get that to work in the lab setting. And so she helps move that work on, even though she's not an author on that publication, she's noted and acknowledged for her contribution along with other undergraduates. So she gets a degree in chemistry from Pomona, and then she's going to move, and her father again pushes her to apply to other schools, and she applies to Harvard and she gets in. So
Hey,
1985. Yes?
I'm so sorry to interrupt you, but we're not seeing your slides advance.
Oh,
So
is that, you don't see them at all advance?
we see your title slide, but nothing's advancing. There we go. Now they're advancing.
Is do they not advance now?
It looks like they'll advance every time you hit the arrow key, I believe.
Oh, so I can't do it from my computer. I have to do it from the arrow key?
Not necessarily. You can do it however you would normally do it, but we're just not, they aren't advancing at this point. We are seeing your second slide now, the title of it.
Do you see that now?
Uh-huh.
Okay. All right. So I was just seeing that. I haven't talked. Maybe it's because I hadn't advanced it yet. Okay.
Thank you.
But you see that picture now.
We do of the graduate advisor. That's what we're
Yeah.
seeing.
Okay.
Perfect. Thank you.
Oh, no problem. Thank you for mentioning that. So when she gets to Harvard, Harvard has in the graduate program at Harvard, students do rotations. So they spend a short time in the laboratory of different scientists before they choose a thesis advisor. And she does several rotations. They sound successful from what she reports to Isaacson. And she ends up in the lab of Jack Szostak. And he's an interesting and dynamic researcher. And it turns out that he's going to shift from working on DNA to working on RNA. Now, RNA, ribonucleic acid, is a very versatile nucleic acid, and it's similar to to DNA in that it has a ribose sugar, and it has a base, and it has a phosphate linkage. But RNA differs because it has an extra hydroxyl group, which is going to make it a little more active, reactive, and it's got one different base of the 4 bases that it uses. So it's similar to, but not identical to DNA. And he's changing over, and I'll tell you in a minute why that is. And Jennifer thinks of RNA as kind of a dull, bureaucratic functionary that takes information from DNA and allows it to be converted into a protein, which does the interesting things in a cell. Because that's how we teach about RNA, and that's especially how we taught about RNA at the time she was in graduate school. But it turns out RNA is a lot more fun than DNA. And I just showed this picture. This is going to be one of the RNAs that she's going to work on here. And I just want you to see, I hope you can see my cursor, that it has not just a simple helical shape that has 2 helices wound around each other, so double-stranded like DNA. It has double-stranded regions, but it also has single-stranded regions, and it forms these stems and these loops and these little loops and these branches, and it has this really quirky and really interesting shape. And what makes that shape even more interesting, and why Jack is interested in this, is that her project and his interests are going to initially involve work with a newly discovered type of RNA, which acts like an enzyme. So you take the ribo from RNA and you add the back end of enzyme, and you end up with a ribozyme. And ribozymes were discovered a few years earlier. And those are RNAs that have the property of being able to cut a little bit out of themselves, RNA. So they can take a section out and they can repair the 2 ends. They can put the 2 ends together that were separated and ligate them together. And that takes a non-functional RNA and makes it a functional RNA. And that was cool. And what Jack Szostak is interested in is he's interested in the molecules that are going to create life, the ones at the beginning of time that started life. And DNA doesn't look like it's going to be the molecule of choice here, but RNAs could be. And the fact that RNAs have these catalytic properties make them even more interesting and make that a more likely proposition. So she is going to end up doing a lot of biochemical work to really understand the properties of this particular rhizozyme and others later. And then she does something else that she's going to do at various places in her career. So she's not just understanding how it works, but she's going to be putting in some new features to re-engineer it so that she can get it to do a slightly different function, to copy a template. So that's even more interesting, that now you can actually re-engineer this to do a slightly different function than this molecule is capable of doing. And the other thing she gets out of the mentorship with Jack Szostak is this idea that what you want to be focused on and what you want to try for is to ask the big questions. And so she is asking big questions, he is asking big questions. And the other thing she learns is it's best not to follow the crowd, to embrace risk. This project is a really risky one. It didn't have to work. She didn't have to be successful, but she has really good hands. She's good in the lab. And so she makes a lot of progress. And the other thing that happens to her as a graduate student, she gets to meet Jim Watson. Because Jack Szostak couldn't go to a conference at Cold Spring Harbor, he asks, and Jim Watson writes a letter to Ms. Doudna, because she's not a PhD yet, and asks her to come and give a talk at this conference. This conference will have almost no other women, very few senior women in these fields at this time, very few women in the audience, maybe a few students, maybe a postdoc. But she goes, she gives a talk, she does a great job. Jim Watson is complimentary to her, and then the person next to her, who we'll get to meet in a minute, tells her, good job. So she rises to the challenge as a 23-year-old mid-stage graduate student. Very impressive. You know, it looks like she's got a great future. So she gets her degree in 1989 from her graduate degree in chemistry, and then she spends a couple more years in his lab finishing up the projects. And she realizes that for her to understand the function, what she has to do is learn what the structure of these RNAs are, so she can really understand how they work. And to do that, she goes to, in 1991, she goes to the University of Colorado and she works with Tom Cech. So Tom Cech got the Nobel Prize for the ribozyme, the discovery of those ribozymes in 1989. And he's the man who said, good job to her after her talk at Cold Spring Harbor. And he's gonna set her on the course. He's doing these kinds of studies. And so she is getting started to understand what the three-dimensional structure of these ribosome RNAs are. And it's a tough job. It's a hard job. These are big RNAs. It's the process of how you have to work to see the structure. You have to do X-ray crystallography. So she's following in the footsteps of Rosalind Franklin, and she has to be able to crystallize them. She manages to get them crystallized, but she can't get them stable enough to survive the barrage of X-rays. A visiting scientist suggests she come to his lab in Yale, where he knows how to cool down these kinds of structures and stabilize them so they can get a good structure. So after these two advances, she says, we knew that we had crystals that were ordered enough that we would eventually be able to solve the structure. She is not able to do that as a postdoc because she's going to move on to her first academic job. And she's going to move on as a single person because she had been married when she moved to Colorado, but she and her husband discovered that they weren't exactly in a line. There's a problem here. She really wants to focus on her research in her lab, and he wants to have a life that includes riding his bike and hiking the hills in Colorado. So they part on good terms, but it's clear that they are not well suited to each other. So she starts her job at Yale, and in really that first year and a half, she has success. The student that was helping her in Colorado moves with her to Yale, Jamie Kate. He's going to help by finding a way to infiltrate some metal ions into the crystals to help stabilize it further and give some more information to the X-rays. So they take this two-dimensional structure you see here, which is kind of interesting, looks like plumbing, and then they form these beautiful three-dimensional structures here. So she has success. She has an important paper in Science. She continues to do more studies in that way, and she moves up the ladder. So she's an assistant professor starting in 1994. By 2000, she's a full professor, and she says, now we can see how, I have to move my little thing here a little bit, just a moment. Now we can see how an RNA molecule can form itself into a complicated three-dimensional structure. And she says at the end, one possibility is that we might be able to know enough to cure or treat people with genetic defects. So she's already thinking about how you might be able to turn this basic science into something with an application. So she's going to continue there. Her husband, Jamie Kate, They get married. He's got a job at MIT. She's at Yale. That's a pretty big commute. So they're looking to find a place where they can be in the same institution or in the same town. Yale works hard to keep her. Harvard considers an offer, but she gets a call from the University of California, Berkeley. And eventually she and her husband moved to Berkeley, and now they have a baby. All right. So here she is at the end of her career. I spend this time because I want you to see that she has both the biochemical understanding of the biochemistry, understanding of the structural biology. She's well positioned for the kind of work that she's going to be moving into in Berkeley that's going to put her in line for being able to collaborate with the people is going to result in her winning the Nobel Prize. So I'm going to take a break from that. I'm going to take a few minutes to talk about CRISPR because the book is going to shift to CRISPR. It's the main focus. This is going to be the gene editing tool that's going to be revolutionizing science. So what is CRISPR and when was it discovered? Its first report that is known is in 1986. A graduate student in Japan has been sequencing E. coli, a common bacterium, and he finds these sequences in the bacterial genome. So these two lines represent the bacterial DNA. He finds a series of repeats. So they all have roughly the same sequence. And he finds in between those repeats, small sequences he calls spacers that have different sequences. Doesn't know much about them. They're all in this kind of space, similar position, and he just reports it. 1990, working in a different prokaryote, archaea. These are prokaryotic organisms that often live in very toxic environments or hot environments. They're very interesting prokaryotes, not the same as bacteria, but prokaryotic, meaning they don't have a nucleus. This is Francisco Mojica. He's at the University of Alicante, and he's a graduate student. And he's, of course, younger than this picture when 1990, but this is the picture I found of him. And he discovers them in archaea, and now, because genomic sequencing has become more common and more people are using it, there are these databases in the United States and in Europe that have sequences of bacteria and sequences of viruses, and he finds these sequences in many bacteria, in many archaea that are sequenced, more than you might imagine. And he also discovers that the spacers here are actually sequences from viruses. So each of these different spacers come from a different virus, and those viruses are known to attack the particular species of bacteria or archaea that he finds them in. And he gives this structure, this segment of DNA, a name. And it has a nice acronym, and it has a clunky, it's an acronym of a clunky set of terms. Clustered, regularly interspersed, short, palindromic repeats. Whoops, getting ahead of myself. And it's called CRISPR for short. And also coming around this time is people looking around in the genome nearby CRISPR. They find these other genes that encode proteins, and those are called CRISPR-associated proteins, Cas, but nobody really knows what they do at this time. And he has a hard time getting this paper published. He is going to make the supposition that these are a bacterial immune system, but he doesn't have any evidence for it yet. And so he has a hard time getting that paper published. But why are they there? And are they really what he said? Well, it is known that bacteria are under attack by viruses all the time. They're called bacteriophage. And you can see this poor bacterium, which is here, has this halo of tens of phage attached to its surface, all injecting their DNA into the poor bacterial cell. And so this notion that the CRISPR-Cas system is an adaptive immunity for bacteria is appealing, but how do you get the data for it? And that data comes from the studies by these two scientists, both French. One is working at Danesco, which is both are working at Danesco. It's a small Danish company. This Danish company makes starter cultures for dairy products like yogurt and cheese. One of them is working in Madison, Wisconsin, and one of them is working in a small town in France. But this company the bacterial species that they work on is a form of Streptococcus, and they have cultures of it going back to 1980s. And so they have this huge record of these bacteria, and they can show that CRISPR spacer sequences, all of those they change with different viral infections. So they're reactive to whatever viruses are infecting them. They also show by knocking out those associated genes that the Cas proteins are essential for the resistance that bacteria show. And they can, by experimentally adding spacers, they can confer resistance to bacteria. So they can actually vaccinate their bacteria against these viruses. So cool. And they're going to get the first patent for CRISPR-Cas9 actually in 2005. And they do that. They are vaccinating their bacteria so they don't get killed off by those viruses. And they write, here we were, workers at an unknown company, sending a manuscript on an organism that no scientist cares about. To get it reviewed was amazing, and we got accepted, and it went into Science. So they're the ones who demonstrate that this CRISPR is an adaptive immunity for bacteria. But how does it work? So the Doudna lab has been working on other RNA and protein systems involved in gene regulation. And so they're well positioned, and now they're in Berkeley. So she's been at Berkeley for a while. And a colleague of her, Gillian Banfield, who works on these bacteria of mines, ones that are comfortable in acidic and kind of toxic environments. They have these CRISPR clusters, and she's really interested to understand how they work. And so at the time, people thought that they were going to work by this RNA interference, by an RNA mechanism. And so Doudna, she's the expert on RNA structure and function. So she's the person to ask to become to collaborate on this. And so she starts her lab working on CRISPR-Cas9, but she doesn't have anybody to work on it. But in walks a prospective graduate student from Montana State, Blake Weidenauer, and he really is interested in CRISPR. He's been looking at bacteria, but now he wants to be able to understand CRISPR and how things work. So she's got that. She has a postdoc who's been working on this other project, Martin Yenick, and he is good. He knows about how to do these things, so he comes on board. And another new graduate student comes in, and she starts to work on these systems. So she changes gears. She starts to work on this system. As she's getting started, she's also going to be pulling together small conferences by other researchers who work on CRISPR. So 2008, they have one, and in subsequent years, they have them. So she's at Berkeley. There's quite a few resources. You can start these little conferences, and these are just informal conferences. People come and talk about the work they're doing, some of it unpublished, and everyone's expected to be good as citizens and treat people's data in a confidential way. But it's a way to share and learn things. And one of the things they learn here is that actually CRISPR doesn't work with an RNA mechanism. It works on DNA. So it's going to be messing up DNA. So she's working on this for a while. And then in the next few years, she's going to be and then they're going to start to collaborate with Emmanuelle Charpentier. And Emmanuelle has a lot of the same philosophy about what she's interested in science. She wants to create knowledge, not just learn it. But she has a different background in the sense that she's been studying bacteria a lot. She was trained as a graduate student at the Pasteur Institute. She's had several postdoctoral positions. She moves frequently. She's has a few years, so this is about 2007. She became the head of a microbiology and genetics laboratory at the University of Vienna. And about the time she's going to be starting to work with Jennifer Doudna to set up their collaboration, she's starting to move to a new laboratory that she's establishing at Umeå University. And what she's going to bring to this collaboration, because now we're going to move into this collaborative, this intense collaboration that they're going to have over a very short period of time. Time has moved on a little bit, and I see our time is moving on too. So I'm trying to move quickly here. Is her small team working at Umeå and Vienna showed that there were really three components to the defense of one of the particular CRISPR systems called CRISPR-Cas9. And that is going to be those three components is what they're going to start to try and understand over the course of about a year and 14 months. So they're going to be working in this period on the structure of this CRISPR-Cas9. And the main players are going to be Emmanuelle Charpentier, Jennifer Doudna, Martin Jinek, and Christoph Krischlinki, who's Emmanuelle's postdoc. And the four of them are going to be working collaboratively to figure out what is going on. And their big reveal is going to be how this system works to cut DNA. So Cas9, that's the protein, that's kind of this blue lump back here. It's going to make cuts. So it has two cutting zones in it. It's going to make a cut in this strand and a cut in that strand. And there are two important small RNAs. This CRISPR RNA is going to be what's needed to detect the DNA sequences that are the target sequences. And the tracer is going to activate that CRISPR RNA, and it's going to hold on here to help it find the target. So the tracer has a really important function, not just the CRISPR. So the tracer connection to the function of this CRISPR-Cas9 gene cutting system is one that other groups are not going to quite understand quite as well as this collaboration. And the other advance they made was to show that they could take these two RNAs, combine them into a single RNA called a guide RNA, and that guide RNA could be engineered to cut any DNA. So it can be used in any different system. And their paper comes out in, it comes out in Science in June of 2012. And they put this all together, get it submitted to Science just before another one of these little conferences. The two postdocs are going to give this talk for this little conference of CRISPR workers. And it turns out there's another group there that's going to come and talk as well. Virginia Sickness is now a professor at Vilnius University in Lithuania. And he also was very close to getting exactly the same thing. So they have these competing talks at this little conference. And his paper is going to come out in September. It's very much the same thing. And you'll see the other two authors are the Danisco scientists, the ones that discovered the immunity properties of CRISPR-Cas9. But he writes, Jennifer's paper was clearly so much better than ours. It wasn't close. It was the tipping point that moved the CRISPR field from an idiosyncratic, interesting microbial world feature to a technology. So the next phase of this story is going to be gene editing. How do we use this CRISPR-Cas9 system? So I have a little video, which I'm going to try and play, which I'm just trying to get set up here. Just a moment. Maybe I'll see if I can't show it this way. Anyway, it's just a short 1 minute video that shows you how this can be used to edit DNA. Because editing is what scientists are going to want to be able to do. Oops.
So we're not able to get any audio on that video
Okay.
from that view. Do you want me to share it?
Let's move on.
Okay.
So I couldn't show you that, but you can see it. It should be in the materials that come afterwards. But the point is that you could use it to do anything, and you could add different DNA, and you can make excisions that you can remove DNA. You can make deletions in them. And the genome editing race is on. And astonishingly, that the original science paper was in June of 2012. By December of 2012 to January 2013, 5 groups have papers on that. They're going to be out of Harvard, Broad Institute, MIT, Berkeley, and then there are 2 other groups. They all do the same thing. They show that they can insert the CRISPR-Cas9 system into mammalian cells, human stem cells, and edit genes in the nucleus. And I'm running out of time to talk about that in much detail, but this is going to set up some conflict between the Broad Institute MIT, where Ben Zhang is working, and Jennifer Doudna on who has the priority here. So their patent wars are starting. This is a big deal. The ability to easily modify mammalian DNA is a tremendous scientific advance, and it's something for which those institutions could stand to make a lot of money by licensing that technology to others. So that's going to create a patent war. That war is still ongoing. They had just some more hearing about that this last week. And these individuals are going to start commercial ventures for those licenses with their universities. So there's a lot of elements of conflict in this part. The book talks about this in quite a bit of detail and all of their different contributions. It's, it's a place where there's a lot of competition. It's difficult to manage. They do a pretty good job, but it's a difficult management for everyone. It's very demanding. So once you have this possibility of doing this genome editing, what kinds of things can you use it for? And he talks about a couple of those things. One of them you might expect, there's therapeutic uses. In this case, this woman's blood cells were modified to produce fetal hemoglobin, reinserted into her body after they had been extracted to help cure or to treat sickle cell anemia. So in this picture, she's back. She's 9 months without symptoms. So that's a therapeutic use. A variety of them are in process and testing. That's a use on somatic cells. That's important. And the other is one that maybe you wouldn't have expected to have happen. Biohacking, do-it-yourself genome editing. So Joshua Zanier he's a PhD, he's had a postdoc, he's a skilled molecular biologist, and he is selling kits to genome edit bacteria and frog eggs and things like that. So this is a technology that's much more accessible than previous technology. And so it's something of a concern, and it causes Jennifer Doudna, even after the papers come out in 2012 and over the next few years, to think about how do we navigate these rules. Scientists have traditionally, since the 1970s, when they were faced with the problem of recombinant DNA, they hold conferences and get together and think about how they can regulate themselves to control this new technology. In this case, genome editing of the human genome is the most prickly of those issues. And the goal here is generally to find a prudent pathway forward for the possibility of keeping this going, especially if it's safe and medically necessary, or to call a moratorium until moral and ethical issues are resolved. On the society side, a lot of that happens with scientists because they get asked to testify in front of congressional hearings. Here's Jennifer Doudna testifying in front of Congress. And there are issues of cost, justice, safety, value. So there are different ways that both scientists and society try to navigate these rules so that technology doesn't get used for inappropriate or difficult purposes. And that was all well and good. So there were several of these conferences. And then just before a conference that was happening in Hong Kong, Jennifer gets this email that says baby's born. It's from He Jiankui from Southern University of Science and Technology in Shenzhen. And he's telling her in this email, a few days before this conference, another conference on the ethical use of this technology, has reported that 2 girls have been born after their genomes were edited and implanted. So 2 embryos were edited. He is fully prepared to have this be a public announcement. At some point, it kind of gets out of his control. He submits 5 YouTube videos. You can go get this off of YouTube, where he describes the process by which they have done this and what they were doing. Jennifer and others are appalled and disgusted, and they're trying to figure out how to manage he's coming to their conference. How are they going to manage this conversation? Are they going to have him talk? What's going to happen? It turns out the editing process was basically a hack job. The babies didn't get the edits that he had claimed that he was trying for. There are a host of problems with how this study was done. Although must have had support in China for this work because he had to go to a hospital to use their IVF technology. In the end, because of the disgust and the controversy, in 2019, he was charged. He pled guilty to illegal medical practice. He was fined in jail. This is sort of the worst case scenario for how things might happen. And as a result of that, the end of this book asks a series of moral questions. The lead off to this chapter is, if scientists don't play God, who will? Another provocative statement from James Watson. And it brings up who's going to be the ones to make these issues. And so Isaacson talks about what kinds of diseases are the ones we should treat? What characters should we modify? Should we enhance, and what physical attributes to enhance? And he has a series of examples that he brings in there. And then the question is, who decides, individuals or the community? So that's where we're going to leave that issue. And just to end with the fact that they get the Nobel Prize in chemistry, the first time 2 women have been standalone winners out of not very many women who've gotten the Nobel Prize, and for their work on the structure and function of that CRISPR-Cas9. And because we're kind of out of time, I just want to say, I think that he did a really good job of showing how a scientist is created. Jennifer Doudna is an exceptional scientist. She takes risks, she's very good, she's collaborative, she's thoughtful, she's active, and she's been lucky. People come in, and that luck is the luck she's created because she's been in a very good position to do this kind of work. And I think it also shows how science has grown from this very small few people who are doing it to this really big enterprise. You see all these people involved in generating the science. many more women. When she was and I was at Cold Spring Harbor, going to classes there and teaching classes or having them, very few women. But now there's lots of them. She has a lot of collaborators who are women. And I think it's really a testament to a great research career that's still continuing. And she has the moral interest and solid foundation to ask, how should we be moving this forward? And so she's moving the ethical questions forward as well. And so I think that's really an important, something that's important about the science and the science that she's done. So I'm going to stop there, and I'm going to pause showing my screen. And I guess I would like to take any questions.
Great. Thank you so much. That was really interesting. I've learned so much. We do already have a question, but I do want to invite audience members now to please be submitting your questions and we can take those. We have about 8 minutes left to get to some of these questions. So one of the questions we've received already is from Randy, and they ask, why doesn't CRISPR and mRNA lend itself to adenovirus vaccine?
You know, it's you could use you can use, I'm not sure I exactly understand the question. It's thought that you can maybe use CRISPR as a I'm not sure how to think about that. Adenovirus. So one of the, I'm not sure I'm going to be able to exactly answer that question. One of the ways that you can get the CRISPR components into a mammalian cells is you do use viral, the viral shells of something like adenovirus to actually get those into cells. That's part of the way that they're able to get that machinery into a mammalian cell to change the DNA. I don't think that's the question you're asking. I'm not exactly sure how they would use CRISPR to generate a vaccine. So I'm not sure I know exactly how to answer that question.
No problem.
Sorry.
Thank you. Thanks for trying. Ruth asks, what is Doudna doing at this time?
So she is continuing to study those. She's writing a lot at this time. I think she's managing a pretty big lab. I think she's managing also these aspects of the use of this technology and how it's being used. So she's doing a lot of outreach in that sense. They're continuing to work on making more progress on genome editing in mammalian cells because that's part of what her lab is doing now.
Great, that's really interesting. That was all the questions we had, but I want to give maybe a minute to see if anybody else wants to submit anything. Barb, what is kind of the most fascinating thing that you learned, or what did you most get out of reading this book and doing this review?
I think the most interesting thing for me was the whole history for how, in fact, those CRISPR sequences were discovered. I mean, because of my field and because of the places I've done in the science I've been involved in, the reading I've done, you know, I knew about the gene discovery and genetics, and I knew a lot about the conferences that they had for all of those parts of it. But learning how CRISPR was discovered and how it was and how they manipulated it, and the role of these these guys working at this Danish company. I mean, who would have expected, right? And their enormous contribution to understanding how this worked. That was for me really fun, that discovery process. And
Yeah,
we graduate students at the same time. She's doing better, but you know, it was nice to see that she progressed in such a way and had such a great training and career.
yeah, definitely. Thank you. Oh, we got another one come in. Let's see. Carrie asks, I would like to know what, if any, is the role of government oversight in the field of gene editing work, and which agency might be involved in doing this oversight? Good question.
Yeah, so good question. And I didn't, in fact, gene editing, all of these things are under the purview of granting agencies largely. So when you have a grant and those agencies have rules about how you do these kinds of studies. They've had them for recombinant DNA for a long time. You have certain rules that you have to do and certain kinds of record keeping you need to keep about what's in your lab and how you're doing it and what you're doing. So to get the funds to do this kind of work, it's not cheap. This is expensive science. That's the other thing I should say. It's not cheap science. And so you get grants from various private entities or foundations and federal government, and those have rules for that. NIH puts out rules for those so that you can't do certain kinds of studies on human embryos, for example. This CRISPR doesn't change that. Private foundations are often outside of that umbrella. If they don't ask for federal funds, they're not necessarily operating under the same rules. So NIH is important for that. I think one of the things that she's and those congressional hearings are, how's the best way to regulate this funding? NSF, NIH, those kinds of science foundations are how those regulations are typically put in practice for practicing scientists. It wouldn't stop, I believe, any private individual from choosing to do those things. IVF clinics operated independently of any oversight for decades because largely because we chose not to allow those things to happen under a regulated system with scientists. We let that go into the private sector. So good question.
Fascinating. And then we got a whole little flurry of questions now, and these two are related from Scott and Mark. Is CRISPR technology a factor in the production of mRNA vaccines? And Mark asked, was CRISPR used to create the COVID-19 mRNA vaccine?
As far as I know, no CRISPR technology was not used for those purposes. It's certainly possible that one could use CRISPR RNA in some way to modify existing DNA in some way to create a vaccine. It's certainly possible that I think there are also alternative ways to do that, so you wouldn't require CRISPR for that particular job. Not as far as I know, is what I can say.
Great, thanks. You may have covered this actually a little bit in one of the questions we just asked about government regulation, but John asked, are there any ethical restrictions on current research that maybe we haven't talked about yet?
I mean, there's current restrictions on human embryo research and the types of cells one can, on embryos themselves or human stem cells. So there are restrictions on the kinds of work that can be done with those. I believe you cannot work on any embryos that would then be implanted into someone to have a live birth. There are and there are restrictions on the kinds. I mean, there are existing restrictions on the kinds of sort of bacterial or viral entities you can work with. Those restrictions are covered under, you can't really work on smallpox virus outside of certain very special places. I think now not at all. It's maybe not a good example, but there are special lab requirements that you have to make to work on certain, like if you wanted to say dengue virus or Ebola virus. So there's all of those restrictions that exist. So if you wanted to use CRISPR to modify those viruses or toxic bacteria or something of that sort, those are already pre-existing regulations that would affect how you use it. CRISPR is just the tool. And so I think those would still be in play for those kind of things. So maybe that is something that helps you think about those already existing regulations for the entities you're going to modify.
Yeah, that's really helpful. Thank you. It is just 1 o'clock, just a little bit after 1 o'clock. So I think that's probably all we have time for today. I'd like to invite Connie, Deborah, and Dan to join us again just for a proper farewell to say goodbye to everybody. Hey, y'all.
Hi. Oh, thank you so much, Barb, for taking us inside that fascinating world. And thank you, everybody, for joining us.
Well, thank you for having me. I enjoyed it. It was fun. Thank you.
Great.
Well, then we'll say goodbye and see you next time.
Thanks,
All right.
everybody.
Bye, everybody.
Bye.